Hello, in today's lecture we'll be talking about imaging artifacts. Now, what are artifacts? Like any other medical imaging modality, ultrasound also has its own artifacts. And artifacts are unwanted features in the images that don't actually represent the target of interest. And it can cause false portrayal of the anatomy or function. For instance, can cause missing structures to not be seen. It can cause mislocation or distortion of objects. Can cause also appearance of fake objects or features that wouldn't have been there. As well as for in terms of Doppler, it can cause calculations of incorrect flow speeds. And what causes these artifacts? Broadly speaking, artifacts can be characterized as a type of noise, but noise is typically random. Image artifacts, on the other hand, are deterministic, and it can occur when assumptions of image reconstructions are violated. It can also be caused by any technical or human errors or hardware malfunction that can cause also these artifacts. There are also several types of artifacts that exist. It can be physical artifact, it can be also a system independent type, or depending on the operator. Let's go over some of the common assumptions in image reconstruction and when violated can also cause artifacts. For instance, one assumption is that throughout the entire tissue of interest, the speed of sound remains constant. So in most ultrasound systems, 1540 meters per second is the constant speed of sound that is in the system. However, as we know previously, that different tissues have various sound speeds, and non-uniform sound speed in the tissue can cause depth location errors, meaning that the distance at which the tissue is actually is not being portrayed in the ultrasound image. Another assumption is that there is a fixed attenuation coefficient, but we know that tissues are not entirely homogeneous. They're inhomogeneous. Therefore, the attenuation through tissues can vary between different types. Another assumption is ultrasound beam propagates in a straight line path. But as we know that there are boundaries in the tissues that are not exactly perpendicular to the transducer, and therefore the sound can either refract or go in other directions that are not being seen in the transducer. Also, an assumption is that only first order or single reflections are present. However, in the tissues, there can be some multiple reflections, meaning that the ultrasound is bouncing back and forth from different tissue structures because of the differences in acoustic impedances of these tissues. Another assumption is that ultrasound beam is planar. But as we have learned before, that the ultrasound beam is not entirely a planar wavefront. There is a finite thickness of the beam pattern, as you would have already learned. Also, no side lobes are present is an assumption, but we have noticed that there are spatial and temporal side lobes present in images. Also, we assume that the transducer is adequately damped, but several transducers might have ringing involved, which can also affect it might cause range ambiguity or difficulties in looking at the time delay of the different structures of the tissue. We also assume that scattering is diffuse. In the previous lecture, we had a lecture on different types of scattering, and one of them is Rayleigh scattering. Now, we've also learned that depending on the size of the object, it can also diffract to the scattering. It can have scattering patterns that are not entirely isotropic, going different directions. Also, we assume that all transducer elements have equal sensitivity, but that's not necessarily the case with electronic instruments. Time gain compensation is also set properly, and we've learned that time gain compensation is an operator-dependent type of parameter, and it requires expertise of the sonographer to be able to dial those TGC knobs appropriately that will help visualize the tissue structure properly. Also, we assume that no aliasing is present, particularly for Doppler. But if Doppler settings are not done properly, then there could be some aliasing which can also impact the Doppler measurements. The first artifact we'll discuss is what's called the reverberation artifact. It's typically also called the comet tail artifact, and we'll discuss why. So what happens is that if you have a strong reflector, so let's look at this example here. This is an ultrasound image of a needle inside a phantom. And this type of scenario typically simulates a procedure known as needle-guided biopsy. So you have your transducer that will be imaging from the top, and the transducer signal is being transmitted, propagates. The needle has a really high acoustic impedance mismatch towards the tissue. So it's a bright reflector, a really strong reflector. So that reflection from the needle will go back to the transducer. And if that reflected signal doesn't die down, then the signal will then bounce back from the transducer down to the needle and so forth. So it's called multiple reflections. So what you can see here is that there are these multiple reflections due to the strong needle reflector. And as the sound is going back and forth from the transducer and the needle, you can see that the amplitude of those multiple reflections slowly fade away as a function of depth here. What these reverberation artifacts can cause is also false targets that can be seen in a reconstructed image. So this is a source in which there could be some target that is not there, but is actually a reverberation signal from a strong reflector. The next artifact is acoustic shadowing. And what happens here is when the ultrasound beam encounters a structure that is highly attenuating, then assumed in a image reconstruction. So in this case, distal structures will appear darker than they may seem. So for instance here, this is an ultrasound image of a stone in a gallbladder. So gallbladder is typically filled with fluid-like substance. If there is a stone here, now stone has a really high acoustic impedance mismatch with tissue. Therefore, it'll act as a strong reflector. So you can see here that this is a bright echo coming from the stone. Much of the signal is being reflected back to the transducer, and therefore not much of the signal also gets transmitted through this stone. So you get this shadowing artifact here. Stones don't only cause this effect, bone also causes the effect because of its high acoustic impedance mismatch with other soft tissues. Metal implants also cause this effect, as well as any calcification that occurs in the body. On the other hand, there's another artifact called acoustic enhancement, and this occurs when the ultrasound beam is interacting with a low attenuating structure. In this case, the distal structures will appear brighter than it should be. So for instance here, an example is this ultrasound image of a bladder. Your bladder is mostly fluid filled right here. And as the sound is propagating through the tissue towards the bladder, not much of it is being attenuated. And therefore, a lot of it is being scattered back from the distal part of the tissue. So we'll define now what proximal distance is, distal is. So proximal is close to the transducer, distal is further away from the transducer. So if you have a fluid-filled structure like a bladder or a cyst, then it can cause this acoustic enhancement in the distal parts of the tissue here. Another artifact may be due to the non-uniform brightness or resolution because of beam diffraction. So what happens in here is that, as you could see that for this ultrasound image of wire targets, if you have a focused beam, most of that ultrasound beam is being focused at a particular region. And in this case, the focus is somewhere around here. Here, the echogenicity is much higher compared to the other parts of the beam. Therefore, there's also beam spreading that occurs as you move further away from the focus of that ultrasound beam. And because of that beam spreading, there could be also attenuation of the signal of the echoes that are coming back from these other wire targets that are outside of the focal region. The next type of artifact is called range or ambiguity artifact. So how this happens is, for instance, in this case here, we have our transducer that is imaging a region of interest. There could be a structure that is of interest to us. There also could be another stronger reflector that is outside of our region of interest here. So our transducer is sending a pulse. The pulse interacts with the tissue, and our pulse repetition frequency is typically set such that the echo coming back from the deepest part of your region of interest has enough time to go back to your transducer. Then the transducer would move to a different lateral or horizontal location, and the same happens. If your pulse repetition frequency is too high, so the echoes from the deeper regions do not fully die out before the next pulse is being transmitted. So if you have a stronger reflector right here, what happens is that due to the high pulse repetition frequency as echo is sent, but this echo from the strong reflector doesn't die out, so then it then interacts with the next pulse, and that can cause some ambiguity artifact, as you can see here. It may cause deeper structures that are not initially part of the actual region interest to appear in your ultrasound image. Side lobe artifacts are also there. It can happen in single element transducers, and grading lobes can also happen from arrays. And it can appear as additional beams, additional to the primary sound beam. And it can cause the objects to be displayed incorrectly in the horizontal position of the ultrasound image. So for example, in this ultrasound image here of a bubble cloud, and what a bubble cloud is that there are micro bubbles that are situated in one location here. And in this image, the microbubble cloud is located at the center of image where you would see high echogenicity right here. But as you can see in this image, because of these side lobes, you can see this other echogenic signals that are coming on the sides of this bubble cloud. But this is not representative of the actual bubble cloud. There are also edge refraction artifacts. And what happens here is that, for instance, you have, if you take a look at this ultrasound image of a gallbladder here. Gallbladder, as we know, is fluid filled. But as the ultrasound propagates through and hits an edge of that structure, there can be some refraction at the edges of the object. And those refraction can cause the edges to appear dark. Now, because of the refraction, the beam gets diverted into a different direction, and the signal does not go back to the transducer. And therefore, at that location, there's no signal that's being reconstructed here. Therefore, this edge refraction artifacts also occur. Okay, the next artifact is the mirror artifact. And how this happens is a reflection from structures located close to strong specular scatterers can produce a duplication of the object in the image. To give you an example here, so I am standing in front of a mirror and a camera is taking a photo of me. But as you can see, myself is also visualized in this photo because of the mirror image. So the same thing can happen in the ultrasound image, wherein, for example, in here, in a liver ultrasound image, you can see that the transducer is now propagating down its signal to a certain location here. And you have this really bright echo from a specular scatterer right here. And because of this mirror artifact, the specular scatterer actually acts as a mirror. And so you can see the similar structure that is observed at this region of that image. There's also some issues also with visualizing specular structures in the image. As we have discussed before, typically specular scatterers are not always perpendicular to the transducer face. So what you can see here is an image of a liver, and here is a specular scatterer, which is a diaphragm. As you can see, as when the diaphragm is not exactly horizontal in this image, then the echogenicity of the diaphragm decreases. and that's because of the oblique angle of incidence. So we had discussed oblique incidence when we talked about reflection in a previous lecture here. If we have a boundary, an incident wave is propagating at a certain angle, and the reflected wave will bounce back almost equal to that the angle of the incident wave. And therefore, not much of the incident wave will be reflected back towards the direction of the transducer. And so you can see some parts of the diaphragm that are much less echogenic compared to the parts that are more perpendicular to the transducer propagation direction here. Another artifact is called the transducer ringing artifact. So if the transducer has a high Q factor, meaning that it really rings, especially in the near field, if you have for example, here in this signal pattern here, this is your ring down artifact from your transducer signal, and you have an echo here. If the object is further away, then you can be able to measure the time of flight from the echo and the ring down artifact, this difference. But if your echo or if your structure was really close to the transducer and you have the ring down artifact, the signal, the echo signal from the structure will interfere with the ring down artifact of your transducer. Therefore, it will be difficult to identify the scatterer at that location. So this is what's called a transducer ringing artifact. Doppler also has artifacts, as we would have learned that the Doppler signal helps you assess blood flow velocity in moving structures such as blood. So Doppler signal we know is dependent on the angle of the incident beam and the direction of the blood flow. And typically in Doppler ultrasound, we usually image at angles between 30 and 60 degrees, because if you exceed 60 degrees, then typically the Doppler frequency shift would be inaccurate. But tissue motion can also cause some artifacts in Doppler known as flash artifacts. So if the tissue is also moving, then the Doppler ultrasound will also sense that that is a moving structure. So you can see here in this image of a beating heart, you can see that there is a vessel somewhere in the middle of the image, but there's also some color map signal that's coming around the tissue region. And it's because the Doppler is visualizing this as a moving target. So these are considered artifacts. But not all artifacts can be bad. Some of them are also useful and helps with diagnosing the type of disease a person has. And many sonographers are also trained in understanding these types of artifacts, which will help them with their clinical diagnosis. So an example would be identifying whether there's a cyst or a tumor present. So we have shown previously an example of a cyst, and here I show an example of a renal cyst. Basically, the acoustic enhancement artifact that you see here that is distal to the transducer beyond this cyst, actually, it helps these sonographers identify that, yes, this is a cyst because of this acoustic enhancement artifact. Another example is that if you're trying to detect calcification in tissues, we know that calcified structures have a high acoustic impedance mismatch compared to surrounding tissues. So then you can see that there will be a higher echogenic region that is at the location of that calcified region here. So here's an example of calcification in a liver, and you can see that this bright echogenic signal coming from that calcification, as well as this distal shadowing that occurs because of the presence of this high calcification region. So this distal shadowing helps confirm that this is a calcified region in the liver. Another example is, in lung ultrasound. Typically, a healthy lung ultrasound, you wouldn't be able to image deep beyond the pleural line. But the way ultrasound would help is that if you have a fluid-filled lung or a fibrotic lung, okay, fibrotic means a buildup of proteins in the tissue. What happens in this lung ultrasound is that here somewhere is the pleural line, which encases your lung. And here are regions of where there is healthy lung, because lung is mostly air-filled. A healthy lung would be air-filled. So it would cause some shadowing in that lung. But if there is some fibrotic tissue, then the ultrasound signal would go through the lung and will be able to propagate deep into the lung. So these high echogenic regions here that you would see are called B lines on lung ultrasound. And this was quite useful for determining whether patients with COVID do have that disease, fluid-filled lungs. So in summary, in this lecture, we have talked about different artifacts in ultrasound images, how they are formed, different types, the sources of the artifacts, how they can degrade image quality, but they can be corrected based on some signal processing techniques, and some of which we will discuss in other lectures. But not all artifacts will degrade image quality. Some of them can also be useful. So in terms of identifying cysts, identifying calcification in a diseased tissue. So this ends our lecture. I'm looking forward to seeing you in the next lecture. Bye.